All right, so if you go on your computer and open up your email, you will find a link in your email that allows you to join a Nearpod. Has anyone ever used Nearpod? So it's kind of this cool program that I used last year to do genetics with my class. So we're going to try it out. If we don't like it, we don't have to use it. But basically what it is, it allows you access to my slides right on your computer, and it's collaborative. So I get to see what you're doing, you get to see what I'm doing, and we sort of work back and forth through that. So our next topic is the genetic code, human genetics, what makes you you. So I'm kind of curious what you guys know about that already. So if you've managed to get the code into Nearpod, I see there are 3 of you on there right now. There are more than 3 of us in this class, so hopefully more of you will hop on there. But go ahead. This first slide is a tweet, right? So you get to tweet 250 characters about the genetic code, right? What do you know already about DNA? Right? DNA, genetics, anything. It can be, I know nothing about genetics. I know everything. Here it is. One thing I don't want you to do is Google what is genetics, copy paste it into the box. So ready, set, go.
Can we like look it up? Just not looking it up. I'm sorry, I was zoned out.
Just don't look it up. Just like, for you right now sitting here, even if it's like bare bones, I know that there's this thing called DNA, kind of, sort of, and that's about it. Oh, big word, Kira.
I wasn't sure if it's nucleose or nucleotides or nuclease.
Yeah, nucleotides is right.
Is it right?
Yeah.
I'm like, nucleose, nucleotides. I was like writing the whole time. What is it now?
Cool, right. You can throw someone a like if you enjoy Jackson. All right, so we've got some good things
Can
here.
we just like write words like we remember? Or like we don't know what it is.
Yeah, if you want to write something up there, a word or something that you like.
Oh, no.
Replication, nice.
Yeah.
So, cool. I like these. We've got some good stuff in here, right? DNA makes your body look the way it does, right? It's something about you physically that is encoded in your DNA, right? You get it from your parents, right? So it's passed down. Kira's talking about nucleotides, which are part of the structure of DNA. May knows a little something about dominant and recessive pieces of this DNA, right? It's complex. It makes you you, right? To a certain extent, if we go in and edit your DNA, you are no longer going to retain the same pieces that make you you. And this sort of genetic code, this DNA, is actually pretty simple in terms of what makes up the code. So we're going to break that down today, look at DNA and the structure of DNA, and how that structure of DNA relates to the function of DNA. Okay, so right, human DNA, it has three main jobs. Okay, it does three things for us. It stores information, right? So it stores all this information that allows you to grow, react, maintain, right, everything about your physical body. It's able to copy that information, right, and then transmit that information out, which is really, really important. You can think of human DNA as a book, right? How to be a human is basically what your DNA says, right? Your DNA is stored in the nucleus of each cell, right? So that is your human library, right? It is your place where all of that information is stored. Just like books, right? DNA can be copied and then handed over to something or someone, right? If I have a book, I can easily give it to May. She can read that book and then, know all the information. Same thing with DNA, right? So obviously DNA is not a book, but it does need a structure that's similar to a book, right? Where it can be read, right? In some way, it can be read and translated, right? This word got deleted, but our DNA is made up of components called nucleotides, which was the word that Kira threw out. So these nucleotides are essentially the words in our book, right? They give us information and tell us what that DNA is trying to communicate. And there's actually only 4 different nucleotides that make up DNA. 4 different things that, combined in different ways, give you the code that makes you you. Okay? Does anyone have questions so far? I'm kind of blowing through this. So DNA is made up of these 4 nucleotides: adenine, guanine, cytosine, and thymine. We abbreviate these just by their first letter, right? So AGCT is usually how we'll go about it, but those stand for different things: adenine, guanine, cytosine, thymine. Okay, and they are sort of the major part of this DNA that communicates information. There are other parts of DNA, other parts of this structure. The D part of DNA stands for deoxyribose, which is the sort of sugar part of the DNA chain, so it's another part of the structure of DNA, but it's less consequential than the nucleotide. When we sort of put this all together, so deoxyribose, nucleic acid, the actual sort of DNA part of this, we kind of smush these together. It's deoxyribose nucleic acid is what DNA stands for. Okay, and that's, each piece of this is a structure of DNA, right? They have nucleotides, and the sugar that makes up the backbone of DNA is a deoxyribose. Basically, the structure of DNA is kind of cool. Does anybody know what we call the structure of DNA together? Anyone heard this before? The double helix, right? I have one right here, right? And we call it a double helix because this is actually essentially 2 strands of DNA that have been smushed together to stabilize them, right? The gray piece here is the phosphate groups and the deoxyribose, and then the colored pieces are the nucleotides that are matched together. So this is actually 2 different strands of DNA that, when it's allowed to interact, kind of naturally turns into this cool double helix shape, right? And that is dictated because of the structure of these nucleotides, right? So let's, hold on, before we get there. Right, so if you look on your computer screen, we've got a 3D model here of DNA, and you can actually manipulate this on your screen, I believe. Right, can you click and drag? Or is it just me that can do that?
You can zoom in.
Yeah. So, right, if you check out this DNA, right, those two strands that are curled around each other, right, those are the phosphate groups and the deoxyribose. Those are purely structural, right? They kind of protect the nucleotides. The actual code of the DNA is in the nucleotides, all right? And DNA is tricky, right, because it's very, very small. So for a while, we didn't really know what it looked like, right? We didn't have any real idea of what DNA looked like. And so what people would do, scientists would basically, put DNA in a blender, a sciencey blender, blend it all up, and identify the different parts, right? So we knew pretty early on that DNA had phosphate groups, it had this deoxyribose, and these 4 different nucleic acids, right, nucleotides. But the real tricky part of the structure of DNA is, this is a pretty intricate physical structure, right? How did we figure out that it was the double helix? So part of this, unraveling the structure of DNA, was this guy named Chargaff, and he has a rule about DNA. But basically what he did was he did these blender experiments, right, where he ground up DNA and figured out, what was in them. And he found that every time that, no matter what the DNA was, no matter where it was from, a plant, a mouse, he always found that there was equal amounts of adenine and thymine, and there were always equal amounts of cytosine and guanine. Always, no matter what. There was always the same amount in that DNA, right? So he came up with this hypothesis that in some way, right, the two were related. They were paired, right? There were equal amounts of adenine and thymine and equal amounts of cytosine and guanine, so they might be paired, right? And he was correct. The way that the double helix works, right, is that nucleotides, right, are paired up, and they're sort of delicately bonded together, and adenine always pairs with thymine, right? The red one here is adenine, and the yellow one's thymine. And cytosine, which is the blue one, always pairs with guanine, right? So it makes sense that when you grind this up, right, there would be the same amount of green as blue and red as yellow, right, because they're paired up. And what connects these nucleotides is hydrogen bonds. You guys remember hydrogen bonds? What was special about hydrogen bonds? Do you guys remember? Water, all that stuff. Anyone? Literally anything about hydrogen bonds.
Special properties.
Yeah, they have special properties, right? They are polar, right? So we have some polar bonds here that are connecting these nucleotides. The other interesting thing about hydrogen bonds, right, is that they are really strong, right? But they're not too strong that they can't be broken and reformed, right? So something really important about DNA that helps us copy and transmit DNA information is that these hydrogen bonds that connect these nucleotides can be pulled apart, right? And then they can be reformed, pretty easily. Okay. So we've got these pairs of nucleotides, and they form different numbers of hydrogen bonds. The AT pairing, I could have sworn that was working a second ago. The AT bond has 2 hydrogen bonds in between. And C and G has 3 hydrogen bonds. So which nucleotide pairing do you think is slightly harder to break apart? Yeah, C and G, it has more hydrogen bonds, right? So if you have a strand of DNA, right, say I had 2 strands of DNA, identical in length, right, that were, one of them was entirely AT pairs, and the other one was entirely CG pairs. Which one would be harder for me to break apart? Yeah, the one that was entirely CG, right? So the content of the DNA strand, whether or not it's more higher percentage AT or CG, dictates its integrity, right? How hard it is going to be to break it apart. Okay, it's up here.
So C and T can never bond together?
No, they cannot.
But then it's always the same, right? Like it's always AT, CG.
Right, so that ensures that the pairs are the same, right? So that there's some, right, if we were to break these 2 strands apart, they'd be in some way identical in the way that they're paired. But there are millions, if not billions, of combinations of ways that we can order them within the strand, right? It can be, G-A-C-T, it could be A-G-C-A, it could be C-G-A-G-C-G, right? On and on and on. And this is, there are billions of nucleotides in your DNA, right? Billions, if not billions. So the combinations of nucleotide orders within the strand of DNA are literally endless, right? Which is, even though it's only 4 letters, right? It's pretty cool. You can have lots of different combinations. Okay. Questions? Oop.
Those don't ever break, though. When you say break.
So they pull apart, right? So this is sort of the interesting thing about DNA, is we tend to think of, right, when we learn about DNA as, kids or whatever, we tend to think of this as like one DNA molecule, right? It's actually 2 strands of DNA, right, that form a ladder. And it should, in a perfect world, break apart easily, right? This is how you read the book, right? Right now the book, if they're bonded together, the DNA book is closed to protect the information on the inside, right? When something comes along and needs to read that information, it opens the book and pulls it apart and can read, okay, this is, C, T, A, right, all the way down. And then when it's done, right, the book closes back up to protect the information inside. Does that make sense? Does anybody know the name Rosalind Franklin? Anybody know that name? Rosalind Franklin. So there's kind of an interesting story about the discovery of the structure of DNA. So Chargaff made this rule that he discovered about the nucleotide pairs, but it wasn't until much later that we figured out that it was actually a double helix, right? That they were 2 strands paired together and that the helix rotated around, right? So these 2 guys, Watson and Crick, right, were working to try to understand the structure of DNA, and they had Chargaff's rule, right, so they could, figure out what that was and, knew about the nucleotide pairings, but they didn't have the full picture, and they were working in a lab that was taking x-rays of, you couldn't just, leave it on my desk.
I tried. Oh, I tried. It came in this door.
That door's unlocked.
This door's locked, but that one is, right? This one's locked.
Yeah, I'm not there.
Yeah.
Thank you.
Ben wasn't there. I did not put him in for absence because they don't have that ability.
Thank you.
Yep.
So, right, they're looking at basically pictures of DNA. They're technically X-rays, and they don't have the full picture, pun intended. But there's this woman working in the same building in a different lab. Her name is Rosalind Franklin, and she takes this picture right here, which is actually a picture of DNA, okay? Zoomed in really, really small. And essentially what you're looking at here, like this is the backbone, and she's zoomed in to the point where you're, like it looks like an X, so you're looking basically like this. Does everyone see that? Okay, like that, kind of, right? So this picture basically indicates that, right, it shows that there's this helix thing going on with the two strands, right? So Rosalind Franklin has this picture. It gets somehow circulated to Watson and Crick, and it leads to them proposing this double helix model, which ultimately they get the Nobel Prize for. It's a huge thing, but because of women's position in science, Rosalind Franklin gets no credit for her picture. She does not receive a Nobel Prize for her discovery. It only goes to Watson and Crick, and we even talk about the Watson and Crick model, right? That this sort of double helix was their discovery. So kind of I like to mention Rosalind Franklin because she was cool and did some cool stuff for DNA. So, right, so looking again at our 3D manipulation here, right, you can see that they've, obviously DNA isn't colored this way, right, but they've indicated different nucleotides using different colors, right? And it is true, you can see in this model here, right, that some nucleotides are bigger than others and some are smaller. Is it gone? You gonna survive? So C and T are smaller nucleotides than A and G. Okay, so they are smaller, they're different types of nucleotides. I'm not so concerned that you know that, but you do need to know the pairing, right? You need to know that A is always paired with T and C is always paired with G, okay, in a double helix. When I was in high school and I was learning this, there was a couple in my high school, Allegra Terhorst and Chris Gilligan. That's how I remembered it. AT and CG. And to this day, that's how I remember the pairing, which is pretty funny. So if you can figure out something like that, you can probably remember this for the rest of your life. Okay? So AT, CG, right? So given that you know that, I'll skip this for now. So now on your board, right, you should see this little thing of DNA, right? And what I want you to do is draw the matching strand, the paired strand, right? Given what you know, what is that sequence of nucleotides going to be for the strand that I've given you?
Like on the other side.
Yep. So start at the top, try to make it down through the second G. It's hard to draw with your.
You just do the color.
You can just do the color if you want, or you can write the letters. It's up to you. All right, cool. So, right, hopefully you get the gist of this. You know, you're matching the A's with the T's and the C's with the G's and vice versa, right? And the cool part about this, right, is when we peel these apart, right, if you imagine that, peeling these apart, you get two what are called complementary strands, right? They essentially have the same code on them, but in the opposite, right? So where there was a before, there's a T on the other side. Where there was a C, there's a G, right? So in some way, they're related, okay? And that's going to be really important for translating this code into proteins, all right? So I think that's all the time that we have for today. What I would like you to do for homework, I'm going to give you this night to just study, okay? Probably on Tuesday I'll have some light reading on genetics, so keep that in mind, right? Even though you have the quiz the next day, I'm going to give you homework tomorrow, so do some studying tonight to prepare. What? So I'm on duty on Tuesday. We'll talk about that tomorrow. What do you guys think in your pod? You like it? Some people like it. Some people don't like having to have their computer open and their notebook. So we'll continue to think about whether or not we like it.
Thank you, Miss Brown.
You guys.